Thank you very much for the invitation. I'm going to talk about the work we've been doing at Pirbright. We've been working also in Georgia, but today I'm going to focus on vaccines that we've been trying for genotype 1 Benin. And I'm sorry, I'm going to repeat some of the things that the previous speaker talked about, but just wanted to emphasize some things like we know that this is a large virus that encodes many genes, and most of the genes are non-essential for virus replication in cells, but are important for evading host defenses. And that's what we're focusing on in our research. And the rapid virus replication in infected pigs indicates that there is a failure of control by the innate immune system. Again, we know all now about how dramatic the situation is in the world. We've seen several outbreaks in Europe, most of them in wild boar, but we have seen recently also in domestic pigs, mainly in Romania and Bulgaria. And of course, in Asia, they are mainly in domestic pigs. So the question is, why don't we have a vaccine yet? And this was addressed by the previous speakers, but I want just to emphasize some other things, is that there is no conclusive evidence for neutralizing antibodies in ASF, and there are multiple immune mechanisms described already. For instance, in Pirbright a few years ago, we did some experiments deleting CD8 cells, and we proved that they are absolutely required for protection. However, antibodies may still be required, and there is a role for NK cells too. Another important issue is that we don't have correlates of protection, both in vivo or in vitro. And adding to this, there are multiple antigenic determinants that are not fully characterized yet. So we know that inactivated virions do not confer protection, and we also had limited success using DNA vaccines. So in Pirbright, we're trying to produce a live attenuated vaccine as a quick step to find a vaccine. And this hopefully will allow a fine-tuning of safety and efficacy of a vaccine strain. But for that, we need to delete genes that will attenuate the virus and reduce protection against challenge in order to activate innate immunity and then adaptive immunity. However, the replication of this virus cannot be too much that will cause pathogenesis. And this will be also important to have a DIVA marker in these vaccines. And in Pirbright, we're trying to identify such non-essential genes that encode for proteins that inhibit host immunity, and then delete those genes and see if they could be used for a live vaccine. And the most obvious candidates will be the ones that modulate interferon response. And as you know, when the virus infects cells, they will be recognized by the host and by receptors such as TLRs, RIG-IMD5, and CGAS, which is a DNA receptor. And all of them will activate the transcription factors IRF3, IRF7 in P65, then will lead to the transcription of interferon beta. And this interferon beta will act on neighboring cells and will activate the antiviral state, but also lead to the production of cytokines, chemokines, and aid antigen presentation. And we heard already about the MGF family members, and they were being described as inhibiting this pathway. We also had a gene called I329L that is a TLR3 antagonist, and the UK gene that was referred earlier also was recently shown to inhibit the CGAS pathway. You also have A238L that inhibits the pro-inflammatory cytokine production. So we have here already several candidates for vaccines, and we've shown that the deletion of members of the multigene family 360 and 505 from the Benin isolate, in fact, increases the production of interferon by infected macrophages whilst not having an impact on viral replication. And this is similar to what happens when we infect cells with ORT883, which is a natural attenuated virus. And so we decided to delete a different gene, and this is a single gene called DP148R, which we have indication is also inhibitor of interferon. And as you can see in the survival curves in the lowest graph, we could see 100% protection after challenge. Whilst when we immunized with benign delta MGF, we only got 66% protection. However, the problem, as you can see in this graph, is that we have high levels of virus genome copies in the blood. As you can see here in red, we could see at day 5 post-immunization almost 10 to the 6 copies per milliliter of blood. And of course, this is a problem for vaccine development because it can lead to shedding and transmission of the disease. So we looked more carefully at what it was, this viremia or just persistence of the virus in the blood attached to red blood cells. And this question arises because ASF encodes a protein called CD2 that is responsible for binding of virions to red blood cells, I can see in this picture, but also to infected cells. So we reasoned that maybe not all of these viruses that we've seen up to 60 days post-immunization was infectious. And in fact, when we did viral titrations, we lost the infectious virus around day 30. Still, we had quite high titers post-immunization against posing a problem for the vaccine development. So we decided to modulate, to delete CD2, and another option would be to do a different deletion would be a deletion of apoptosis inhibitor. And this would allow the cells to die earlier after infection and therefore reducing virus progeny. And again, ASF encodes several members that are able to modulate apoptosis. We have, for instance, inhibitor of apoptosis A224L. We have A179L, which is a member of the BCL2 family, can inhibit the mitochondrial pathway, and also DP71L, which is able to modulate ER stress-induced apoptosis through modulation of ATF4 and CHOP transcription factors. And so we decided to focus on A179L, and we further investigated its function, and we found that it's also able to inhibit CHOP activation following ER stress, as you can see here. In cells that are transfected with A179L, there is a reduction of CHOP in the nucleus, and also this corresponds to a reduction of luciferase-dependent activation when you have the CHOP promoter upstream of the luciferase gene. And also other groups implicated this gene in modulation of autophagy. So we first deleted this gene from the BA71V, which is a viro-adapted strain, and we didn't see any impact on the viral replication or impact on CHOP activation, which seems to indicate that other genes in the ASF genome can have the same function. But when we delete this gene from BNE97, we notice that this severely impairs replication in the porcine primary macrophages. So this seems to indicate that the mitochondrial pathway is very important for macrophage survival. And then we follow this by animal experiments. And here we use two different groups. We used one group with 4 pigs immunized with benign delta DP148R, delta CD2V. This is an attempt to reduce the viremias that we've seen before. And a group D where we immunized pigs with benign delta A179L. The reason we didn't delete the DP148R from this was because we already had quite a good attenuation, and we were concerned we wouldn't have enough immunity after immunization with this. So we only have a single deletion. And then finally we have a control group with 3 unvaccinated pigs. So first, the temperatures from these experiments, you can see in group C with benign delta DP148R delta CD2V that we had fever around day 5 to 6 post-immunization for 2 to 3 days. But then there was no increase of temperatures after boost or challenge, and this indicated that there was protection against challenge. And the opposite happened with the group D. There were no clinical signs after immunization, but then when the pigs were challenged, the response was very similar to the control pigs, and they had to be culled due to signs of acute ASF. The clinical scores tell the same story. We lost one animal in group C due to other disease not related to ASF. But as you can see, there is no clinical scores after challenge in group C, while in the group D it was very similar to the control pigs. So when we look at antibody responses against the VP72 structural protein, there was a clear difference between groups. In group C, their antibody response was detected from day 10 post-immunization, while in group D, only one pig had an antibody response, and this was just before challenge. Concerning T-cell responses measured by the interferon gamma ELISpot, again, a very good responses in group C that decreased slightly after boost, but very high even before challenge. And in group D, these responses were much lower. Please bear in mind that the scale in the graph, the group D is different. So if you look at the graph down, you can see that there's a marked difference between these two groups. But most importantly, our experiment was done to try to improve our benign delta DP148R vaccine, to try to reduce the amount of genome copies and infectious virus in blood, and we achieved that. So now, if you compare the up graph, which is the one from this experiment, to the one I showed you before, there is a clear reduction of the time that animals have viremia. And most importantly, they still retain sterile immunity after challenge. In the group D, the benign delta A179L, the only difference we've seen in viremias was at day 3, which they are slightly smaller than the animals in the control group. So in summary, when we immunize with benign delta DP148R delta CD2V, we have clinical signs for 2 to 3 days after immunization, and these include fever, apathy, and loss of appetite. But we reduce the persistence of virus in the blood following immunization as compared with benign delta DP148R. Also, we had good antibody responses and high levels of interferon gamma producing cells, and 100% protection against challenge with wild-type virus. In the case of the benign delta A179L, we don't see any clinical signs after immunization, and only one pig had antibody responses with much lower levels of interferon gamma producing cells. We couldn't see any protection and only a delay in viremia compared to the control groups. Interestingly, the pig D4, which had the lowest amounts of interferon gamma producing cells and the highest level of antibody response, was the one that had the highest viremia at day 3 post-challenge. So what we can conclude from this experiment is that there has to be a very delicate balance between virus replication that is enough to stimulate the innate immunity to limit early replication and activate cellular antibody responses. But if you reduce too much the virus replication, then the immunity is not going to be induced and you won't have protection against challenge. I also want to emphasize that, as we heard before, the deletion of the same genes from different isolates does not necessarily have the same impact. And we've been testing these deletions in Georgia, and the results are not the same. And we can conclude that the safe and efficacious vaccines will most likely to have several genes deleted to obtain a balance between the replication and the induction of immunity. And as I've shown you today, we can target different kinds of genes interferon inhibitors, as we know, are very important for the antiviral response of the host, and this is emphasized by the large number of these genes encoded by ASF. And the increased interferon expression will lead to the restriction of the virus replication, will recruit innate cells to clear infection, promote antigenic presentation, and antibody class switching. If we delete apoptosis inhibitors, we will have early apoptosis will reduce the virus progeny, and we've shown that the mitochondrial pathway particularly important for macrophages, as you can see when we deleted the A179L. And of course, other roles of A179L may be having an impact for the phenotype we observed. And finally, cell adhesion molecules such as CD2 may also help to reduce viremias and the virus persistence in blood. And because it's been also shown to inhibit lymphocyte proliferation, it may be also promoting cellular immunity. And finally, I just want to thank my group leader, Linda Dixon, and all the other members in the lab for their support all these years. Thank you for listening.
Very nice. Thank you very much. I have just one question. It appears that in terms of CD2V, you delete it in some viruses and it attenuates the viruses, but in others it doesn't?
It's correct, yes.
Okay, why?
I wish I knew the answer for that, because we have exactly the same thing with DP148R. When we deleted from Georgia, we also couldn't see attenuation. But that's the reason we deleted here in the DP148R background, because our intention was just to reduce viremia, not use that to do attenuation. But yeah, I think that's the million-dollar question.
I have two questions. You discussed the ASF, the mechanisms of ASF persistence, and you hypothesized it's either ASF replication or attachment to red blood cells.
Yeah.
And you didn't give the answer, but knocking out CD2 and the virus going down, and therefore the attachment is less to red blood cells. So it is the red blood cell is the answer, or what is? It's both. Maybe replication in some organs and red blood cell.
I think in the case of CD2 would be the persistence in the blood that we are seeing, and that's our intention was to reduce that, to reduce shedding. But of course it may be due to other things. For instance, with ORT883, which has a truncated CD2, we don't see any virus, well, very rarely see any virus in blood, and still it induces protection, so it must be replicating elsewhere.
Okay. So it's both, thank you. And
yes.
The second question is, we all know, and you elaborated on that if you knock out autologous genes in different strains or genotypes, you don't see the same effects.
Yes.
What are your thoughts on the reason for the rationale behind this?
I think so for the multigene family members, I think it's also a matter of numbers. But of course, because we don't know the function of a lot of genes, obviously they may act synergistically, and if we don't know which ones are acting with each other, it's going to be very hard to predict. So until we don't know the functions, it will be very hard to predict why.
I'll ask a genetics question. So there was a paper maybe 2 years ago that talked about that there was a mutation in NF-kappa B in the African warthogs.
Yes.
So I didn't see sort of inflammatory pathways as some of your targets. Do you think that's too general an effect, or why are you not looking at those?
Yes, so the deletion of the gene A238L, which is an inhibitor of NF-kappa B, did not attenuate the virus. Yeah, I know which paper you're referring. I think it's because the virus has so many mechanisms to modulate NF-kappa B, I found very hard to believe that just modifying NF-kappa B in the pig will have an impact on replication and outcome of disease.
I guess the sort of the larger question is, do you think it's going to be important to test the vaccines on multiple hosts, multiple genetic backgrounds? Because I can imagine that if you have an attenuated virus in the endemic population that doesn't really, doesn't have a strong disease when it's infected, will perhaps not even be able to infect that, and you won't get any kind of protection.
Yeah, definitely, and especially the wild boar, which we don't have the facilities to test with, and its main target in Europe. So that will be very important, and also to use that as by the oral nasal route. We were testing everything intramuscularly, and of course, if we're going to use in the wild boar, we probably will have to use with a bait, and that's also a very important question.
Okay, so we're running a little behind the schedule, so if you have more questions, you can discuss during the break. Thank you.